Yeah. Hello students. Today we'll study about two basic tools of recombinant DNA technology. The first one is restriction enzymes, and you know that these are naturally present inside E. coli cells or bacteria. Now restriction enzymes, these are actually providing natural defense to bacteria. Now, how can it be possible? Let me tell you that there is restriction modification system in bacteria. Name restriction. Restriction means restricting somebody. And because it's a defense mechanism, so it is restricting somebody's entry. Now, restriction modification system is having two parts in it. The first one is restriction enzyme and another one is methylation. Second one is methylation. Now, as I just told you that restriction enzymes are present inside bacterial cell. Now if it is a bacteria and this is bacterial DNA, that is host DNA. So whenever any phage or virus infects bacteria, its DNA is moving inside the bacterial cell. Suppose these are restriction enzymes. Restriction enzymes are also known as molecular scissors because they can digest or cut molecular DNA. Now, what is the specific property? That these restriction enzymes, they recognize some of the sequences of this foreign DNA and after recognition they cut them. So in this way these restriction enzymes, they restrict the entry of foreign DNA inside bacterial cell. Now, restriction enzymes, which are molecular scissors, they are cutting DNA. Now, if they are cutting foreign DNA, first they are recognizing the sequences of that DNA. So, these restriction enzymes, they can cut their own DNA also, but it's not happening. Oh, what is special there that they are not able to recognize? So, there are methylation in their sequences. So, what is the methylation? That restriction enzymes, they fail to recognize their own DNA, their own DNA as methyl group is attached to its one or two bases of the sequences which are recognized by restriction enzymes. So in this way, restriction enzymes, because the sequences they are going to recognize, they are methylated. To these sequences, methyl group is being attached. So these restriction enzymes, they fail to identify their own DNA. So this is the basic property of restriction enzymes. That restriction enzymes, they provide defense to bacterial cell from phages. So it happens in case of bacteriophage. So whenever any viral DNA is entering this bacterial body, its restriction enzyme first recognize the sequence and then cut that, digest that and inactivate that, providing defense to the bacterial cell. But they are not able to cut their own DNA as same sequence are present inside their own DNA, but these DNA are methylated. So every bacteria, they are having their restriction enzymes with corresponding methylase enzymes also. So this is the first very important property of restriction enzymes. Now, next, restriction enzymes are actually of three types, but only one type of restriction enzyme can be used in the RDNA technology. So there are type 1, type 3, type 2 restriction enzymes. Now type 1 and 3 also, they recognize some sequences. But they produce cuts somewhere else. Somewhere else means they are not specific in nature. They are not specific at all. On the other hand, if we check type 2 restriction enzyme, type 2 restriction enzymes, they recognize specific sequences. And cut them and digest them. So because these are specific in nature, so we are using type 2 restriction enzyme in recombinant DNA technology, or you can say in gene cloning experiments. So this is about types of restriction enzymes. Another important feature of restriction enzyme is that when we study about restriction enzymes, especially we are saying about type 2 restriction enzymes. Now these can recognize 4 to 8 base pair long nucleotide fragment, and it is palindromic. Now palindromic means that sequence remains same when we read it from 5' to 3' direction. Now let me explain you with the help of an example. For example, there is a restriction enzyme EcoR1. Now this EcoR1 restriction enzyme, it recognizes a specific sequence, that is GAATTC 5' to 3'. Now its complementary sequences are CTTAAG 5'. So again from 5' end it is GAATTC. Here also from 5' end it is GAATTC. So such sequences are known as palindromic sequences. Now another thing, that restriction enzymes, they can produce two types of cuts. Two types of cuts are produced by them. Like if we check that there is one restriction enzyme, this is 5' to 3', another one is from 3' to 5'. And if we take second example, GAATTC CTTAAG 5' to 3', 3' to 5'. Now there is present line of symmetry in the middle. So this is known as line of symmetry. In all the restriction enzymes which are of 4 to 8 base pair long, line of symmetries are present. Now what will happen if cut is produced? Let me make it again. This is GGCC, or here it is line of symmetry. Now if cut is produced along the line of symmetry, here along the line of symmetry, then we will get 5' GG 3 prime plus 5 prime CC 3 prime 3 prime CC 5 prime 3 prime GG 5 prime. Such ends are known as blunt ends or flush ends. But if restriction enzymes, they produce cuts away from line of symmetry. So in this case it is away. Here it is along the line of symmetry. Here it is away from line of symmetry. Then we'll get 5 prime G plus 5 prime AATT C 3 prime 3 prime C TTAA 5 prime plus 3 prime G 5 prime. Such ends are known as cohesive or sticky ends. Another thing that I just want to explain you about restriction enzymes, that is their nomenclature. So, nomenclature of restriction enzymes. We'll take simple example of most commonly used restriction enzyme is EcoR1. Now here this first alphabet is from genus Escherichia. So E from that, then another two, next two, that is from species coli. So this one is genus, this one is species. Next comes R, this is RY13. This is actually strain. Last is Roman numeral. This is 1. 1, it is actually discovery. Means this EcoR1 restriction enzyme is the first restriction enzyme which is present in E. coli bacteria. So it's clear from here that first alphabet is actually for genus, next two for species, fourth one for strain, and last one for discovery. I think till now we must be aware of all the features of restriction enzymes, that they restrict the entry of virus, then they modify their system by methylation. Restriction enzyme, that is type 2, is used for RDNA technology. They can produce two types of ends, blunt ends or flush ends, sticky ends or cohesive ends. They can recognize 4 to 8 base pair long palindromic sequences. And these are specific in nature and regarding the nomenclature also. So friends, in my next video, we'll study about other tools like two more enzymes, DNA ligase and alkaline phosphatase, vectors and hosts. Till then, bye-bye.